The next session will be on the case studies of multinational and bi-regional Latin American and European partnerships.
Hi, it's a pleasure to be in this workshop, and the next session is a particularly exciting one. It's an opportunity to share examples of existing up and running partnership. The last couple of years has highlighted the crucial role for international collaborations, in particular in the area of research infrastructures. And the following talks represent examples of this partnership from national to regional to international collaboration. Then it's a great pleasure. We are delighted to introduce as a first of these case studies to Kleber Gomes Franchini, who is the director of the Brasilia Biosciences National Laboratory and the Center for Research in Energy and Materials in Campinas, Brazil. And he's going to be presenting one case study in multinational and bi-regional Latin America and Europe partnership. Then, Kleber, if you can join us and show your screen.
Yep, let me try to share here.
Fantastic.
Okay, just a moment. Are you seeing my presentation?
Yes, we can.
Okay, thank you very much, the organizers, for inviting me to present this part of the symposium. We talked before with Ben, and I mentioned that would be more appropriate for me to talk about the infrastructure that we have here and how we organize this to open the infrastructure to users as our center do in general. So I'm part of the Brazilian Center for Research in Energy and Materials. As Harry Westphal mentioned before, this is a center in which we have 4 national laboratories, one for synchrotron, the other one for nanotechnology, one for biorenewables, and the other one for bioscience, as I am the director of the biosciences laboratory. So we are open facility. We help in the strategies of researchers all over the country and also in Latin America and some of users that come from USA or even Europe. Let me show you our strategy. We have the research vision that we can help projects going from molecules to organisms, so we can integrate a lot of techniques and competences that we have here in the center. And these competences, of course, they are not all in the biosciences laboratory, but we, for example, have the microscopy, cryo-lateral microscopy, for example, that is run by Brazilian National Nanotechnology Laboratory, and the beam lines that are located in the Sirius, our synchrotron light source here in Brazil. So we can help and we can support research in the areas of bioscience in which the interest is to decode mechanisms that go from molecule to organisms. So we have the infrastructure to do that, and also the personnel to help this kind of approach. For that, we have a bunch of enabling technologies here, especially the protein technologies that I would mention. The facilities to quality control, the productions, different platforms to produce proteins. We have an NMR facility also, and the possibility to handle proteomics studies also in the infrastructure. But together, we have microphysiological systems, human on a chip, for example, kind of setup, omics, bioimaging that is connected to synchrotron and also to nanotechnology laboratory, model organism units to produce genetically modified animals, a drug discovery unit in which we have a special kind of collection from Brazilian biodiversity, a unit of computational biology, a biomanufacturing unit also that is in charge of producing monoclonal antibodies, and right now we are moving in the direction of producing vaccines also. It's a pilot plant to produce vaccines, and units of tissue engineering. So in any one of these units or tools, we have the ability to help the users in their own strategies. And of course, we have our own team of researchers that do all kinds of researchers in the biosciences area. With that, let me show you some examples, recent examples of integrated projects in which we have we are helping people from different institutes in Brazil, and in some cases also collaborators from different parts of the world. In this case here, I'd like to show you a kind of research that we started with the people from University of São Paulo, in which the genetic departments, they identify mutations in patients, in families that have intellectual disabilities. And once they identify the mutation, we take the genes and make the proteins, and we use protein crystallography to identify what kind of alteration we have in the protein. In this case, for example, just a small change in the structure of the protein, and there is a change in the amino acid here from a glutamine to a glutamate, which is responsible for changing the function of the enzyme. And in this case, we identified the mechanism, and also we went further and produced an animal with a knock-in for this mutation. And we studied a lot of different alterations that these animals had in the brain. For example, there is a reduction in the dendritic spines, in the neurons, an alteration in the synapses, the vesicles that we have inside the synapses, also a spontaneous release of neurotransmitters, and of course alterations in the behavior of the animal. So this is an integrated project that started with the mutation in the protein. So we helped people to understand how this mutation is impacting the structure of the protein, and also we also produced an animal model that reproduces what the human being, the patients had in the case of these mutations. And more of that, we are using synchrotron technology to explore the connectivity of the neurons of the brains of these animals. We are developing together with the serious researchers synchrotron microtomography, synchrotron-based microtomography to explore the connectivity between the neurons and start identifying what kind of problems we have with the neurons that could affect the behavior of these patients, for example. So this is a kind of this is an example of the kind of projects that we can support here, and we can interact with our users using all different aspects of these techniques to help them to go deeper in their own research. More recently, we also have a units of virology here in the lab, and we are interested in solving the structure of emerging viruses. For example, this is a recently published virus in which we use the cryo-lateral microscopy to determine the structure of infective myrovirus. We developed the techniques to isolate the virus, as you can see here on the left. We also have confirmed that this virus would be effective, so they can kill the cells in this plate assay that you see here in the middle. And we solved the structure of the whole particle, viral particle, using cryolateral microscopy. As you can see here, the average resolution here was about 4.4 angstroms. So this is a very nice piece of results that we can produce here in the center, and we can help the users also to do this kind of studies here. As an important step in this study, as you know, a lot of proteins in virus, human virus, can be glycosylated. So it's very important to define what kind of...
Sorry, Kleber, just the alert that it's 2 minutes to go.
Okay, so you can see that we can also define the glycosylation of the proteins. So a lot of different aspects of the results. And here I would like to show you that we have our drug discovery unit that can take projects from the beginning and find with the targets, we can find molecules from our biodiversity, Brazilian biodiversity. We have a collection of more than 15,000 compounds, original compounds, and we can use this in an integrated project with structural biology to define the interaction between the molecules and the targets. With that, I think I can finish my presentation and leave the session to questions. Okay, thank you very much.
Thank you, Kleber. If there are no pressing questions at the moment, we are going to keep going because we are a bit late. But tomorrow there will be plenty of opportunities for questions and answers. And I also would like to invite everyone to join us tomorrow in the satellite event for structural biology, which we will have the chance to discuss all of that. But thank you very much. Great talk. Then next, we would like to invite Manoel Barral Neto from the Fundação Osvaldo Cruz from Rio de Janeiro, Brazil, who will tell us about the International Consortium for Personalized Medicine. And without further ado, Manoel, if you want to share your screen.
Sure. Is that showing to you? I cannot see.
I can see you, but not your presentation yet.
Oops.
Perfect.
Now? Okay.
Now you can, yeah. Perfect, thank you.
Okay. So good afternoon, everybody, and thank you a lot for the invitation. So the the topic that I'm going to present is really on personalized medicine. And I think that is an important aspect is really to discuss with you what is really personalized medicine, because it's not common sense in such a broad audience. But the idea on personalized medicine is really that we can improve the health approach of individuals, knowing better their real situation, instead of using just means or expectatives of based on populational studies. So the idea is really to increase the precision of the approach. And so we are not really totally on the age of personalized medicine. We are in the way to that through increasing precision in the health approaches. So this is why here in America, North and South America, the term that is more used is really precision medicine or precision health. But in Europe, it's the idea of personalized medicine is, I mean, it's used more strongly. But it's really the same concept. So in its beginning, personalized medicine was really genomic medicine, because it was only almost based on genomics. But now it's improving a lot, this approach, in terms of combining several phenotypes and genotypes with different investigations that's possible, like having the whole molecular profiling of the individual, and also medical imaging and other aspects, so all the metabolomic and the molecular profiling, and also lifestyle data. It's important to remember that that health is not only a biological phenomenon. It depends a lot on social aspects and environmental aspects that has really to be taken in account if you want to really understand the health status of someone. So the idea is really to tailor the right therapeutic strategy for the right person in the right time. So the idea is also really to determine beforehand the predisposition to disease and to deliver prevention. So it's quite ambitious, and but I guess we're doing a lot of progress in this direction. So personalized medicine was chosen by the European Union as a top priority, aiming to really provide better care solutions and healthcare solutions to its citizens. And then the International Consortium of Personalized Medicine is really an international consortium that is aimed at improving these relations of Europe efforts on personalized medicine and the global world. So there are more than 40 European and international partners at the moment. In each country is represented by ministries, funding agencies, the European Commission, and so on. So they with the aim of coordinating and foster research to develop and evaluate personalized medicine approaches. And what in the context of today's seminar, we are going to deal more with this aspect of where's the point? Okay. We are going to deal more with the idea of EU-LAC-PerMed. That is really the part of ICPerMed that promotes the interaction with Latin American and Caribbean countries. And in this aspect, there's the EU-PerMed, the funding arm of the effort. And particularly, we're going to talk about the EU-LAC-PerMed, that is really this aspect of international consortium that is more directly related to us here. So the idea is really to that we have to engage like countries in the ICPerMed through this ERA-PerMed. So in Latin America, the South Cone, Chile, Argentina, Uruguay, and Brazil are represented in the ICPerMed, and in the Central America, Panama. And several countries, European countries, are present in this approach. So there is a lot of effort, and Fiocruz is representing these Brazilian efforts in Brazil at the moment. And the idea of the ICPerMed is really to stimulate the community, the integrated community of European and Latin American and Caribbean countries to think and develop ways to really make joint efforts on personalized medicine. We have have been already summer schools with good success, gathering several individuals from different countries for attending it, and it's a kind of a capacity building that is extremely necessary in this field. There are technical workshops that are also of very high quality and has been maintained even in this period of the pandemics through changing to virtual workshops, but we're really able to perform that. And there's this clinical trial help desk that is important in terms of networking individuals, but also discussing regulatory aspects. You can imagine that dealing with human health has a lot of problems in terms of data protection. The European law of data protection is very strict, and there are variations in different countries in these legal aspects, and even in ethical aspects, there are some changes that is necessary to understand. And luckily for us here in Brazil, our law is very similar to the European law in terms of data protection. So we are now, really having more possibilities of collaborating without many legal hurdles in the way. And resources and policies, it's really a mapping exercise that is very important. It's really an important part of the project is really keeping all the information on these interests and efforts and events in this project fundings in the area, and policy briefings, and the best practice and recommendations. So it's a kind of very comprehensive approach to really stimulate both countries to interact. Both regions to interact. And what I'm now going to tell you is that in this field, in personalized medicine, it's a little bit different of what we have been mainly discussing so far in terms of infrastructure. We're not talking only in terms of large or very large physical structures, but we are talking about a whole system that is necessary to develop in terms of fostering collaboration. We can divide some way artificially what is the main scientific pillars of personalized medicine, but I guess nobody will discuss that that we're dealing still with a lot of genomics, clinical research for several other aspects, and now, trying to stimulate.
Sorry, Manoel, this is your 2 minutes left.
Okay, comprehension of social environment, and so it's necessary not only sequencing facilities, but clinical research, large biodata banks, and interoperability and integration. This is a major challenge. And besides that, these ethical and legal aspects and the capacity building that I have mentioned before. So this is kind of now talking about Brazil mainly in terms of this structure. This is the national scientific structure maintained by the federal government as a core. But and this part at the right, that is with this inside this triangle, it's more related to personalized medicine, the laboratory of the DNA sequencing, supercomputer, and the Center for Clinical Pharmacology. But then we also, it's important to mention that we have large structures, mainly in the form of data banks and biobanks. And so these biobanks are really already regulated in Brazil, and our national ethics review board have already approved 56 biobanks in different areas of the country. And these are really, I mean, strictly regulated in terms of also data protection, besides all the aspects of maintaining in a proper way the samples that are stored. And in terms of mainly these data analysis, the Brazilian initiative on precision medicine, based mainly on genomics, this is an effort from FAPESP, but is funded by the São Paulo funding agency, but is receiving material from the whole Brazil, and in order to help implement precision medicine in Brazil. And Fiocruz also has a genomic network that's mainly now, and you see that is scattered all over Brazil, and it's really mainly devoted to sequencing pathogens, not so much men, but is interested in expanding to that in collaboration with the FAPESP initiative. We have very strong links with the FAPESP initiative, and Ishia was a presenter of several of our efforts in EU-LAC meetings. And my last slide is really for talking about this great effort that we are doing in Fiocruz in terms of with the Center of Data Integration and Knowledge for Health that was launched in 2016. And it's really to develop investigative methodologies to work with large databases in human health. The flagship project of CDAX is really a cohort, a virtual cohort of 130 million Brazilians. And we have all these health records, and also several other information that's not directly related to health, but like education and everything, and income, that is really important to health. And several studies are already published with these aspects in terms of evaluating public health policies. It's much more so far devoted to public health, but currently CDAX is incorporating databases of genomics, both human and pathogens, and electronic health records, positioning itself for dealing with precision health in a more broader way. So I hope that I've kept my time almost right.
Thank you. Thank you, and we will continue discussing in the Q&A tomorrow. And we will pass now our change showing the diversity of research infrastructures to the next talk is in plant phenotyping. And we welcome Alexander Morais to Amaral, who will tell us about the Latin American Plant Phenomics Network. And welcome, Alexander, and if you want to share your screen.
Okay, just a quick update on the subject. In fact, can you see my screen here?
Yes, yes, we can. Perfect, thank you.
Okay, first of all, thank you very much, the organizers, for the invitation and for having me here today. I'm here on behalf of the Director of R&D of Embrapa, Dr. Guy de Capdeville. So in the next few slides, I'll be talking about Embrapa, who we are, and how we work, and also our partnerships. I will not focus on plant phenotyping or this kind of infrastructure, but I think, I hope I will give you some ideas about how we can collaborate and how we can do partnerships regarding our programs, so our instruments and so on, okay? Just to clarify, Embrapa is the in-house agency of the Ministry of Agriculture in Brazil. Our mission is to provide research, development, and innovation solutions for the sustainability of agriculture and for the benefit of the Brazilian society. Just to give you an idea, the challenge that we have ahead of us in Embrapa, we are all over the country. You know that Brazil is a continental country, and we are around 8,200 employees, 22 scientists, mostly with a PhD, I would say 90%. As I said, we are a network of research centers. We are spread all over the country, over 40 research centers. Some are dedicated to national thematics. Other ones are dedicated to products. And also a part of them are dedicated to eco-regional topics. Let's say, so there is a research center dedicated to maize, another one to dairy cattle, another one to wheat in Brazil. And we also investigate, let's say, the use of machinery in Brazil, informatics, and so on. We have around 6 research centers dedicated to the use of the biodiversity of the Amazon basin in Brazil. And we organize our projects in Brazil in what we call the portfolio. So we accommodate there all the research projects that we have in Embrapa for our challenge. Just to illustrate, we have around 80 breeding programs in Brazil regarding animals, plants, and so on. And we do scientific cooperation overseas using some tools that we will explore a little bit more after a few slides here. So regarding the research centers, I will show you just 3 or 4 research centers that we have in Brazil. And this is the infrastructure that we have in the north of Brazil. Another infrastructure in another state of Brazil, and so on. Here is our research center on soybean in Brazil as well. And as a matter of fact, our national portfolio covers a number of issues in Brazil, a number of topics. Let's say nutrition, safety, and health, the Amazon, aquaculture. One topic that I think it's particularly important for this initiative here today is the automation, precision, and digital farming. Also meat, coffee, and so on. Tropical fruit, grains, forestry. So as I said before, we are over 30 portfolio of research in Embrapa. Also, organization, innovation, nanotechnology is also a big topic for cooperation in Embrapa, pastures, and so on. As I said, we are responsible to provide solutions in terms of R&D for the big challenge of the Brazilian agriculture. So we produce many technological assets, such as cultivars, animal lineages and races, agricultural inputs, industrial processes, agricultural processes, machines and equipments, softwares, and so on. And of course, we have a way of monitoring the technology readiness or the level of maturity of these assets. Having said that, I can show you a couple of examples of those deliveries, such as the create deep learning data sets and models for apple yield regarding detection and robotics. Also, let's say, develop data sharing protocols, integrate disease data, disease design risk mapping, and biovigilance platforms. Also, we deal with problems regarding big data, develop ICASA data, and it's related to Europe in that regard, the protocols to ensure compliance with international data sharing in precision agriculture. And also in terms of climate change research, the impact of nitrogen inputs in conservation cropping systems, impact of soil conservation practices on soil carbon sequestration, and so on. Well, of course, we don't do that alone. We do that with cooperation, domestically or in Brazil, but also overseas. And we have some very interesting mechanisms to facilitate, to foster those mechanisms, those cooperations. I will just give you some examples here about those mechanisms, but they are basically our LabX program that I will explain in the next slide. But we also have this mechanism that we call the joint calls, and visiting scientists that we can place overseas in terms of cooperation. And also we carry out co-funded projects that we establish with our partners overseas. Regarding this LabX program that I just mentioned, this is our, let's say, the virtual LabX overseas. Basically, we place in other labs overseas Embrapa research leaders in world-class hosting laboratories. The topics for research are basically cutting edge, and we place and we give support those research leaders for 2 to 3 years to work as catalyzers of cooperation for Embrapa. So we've been doing that for over 20 years, very, very successfully in US, in Europe, including in Germany, in the Netherlands, in the UK, and traditionally in France. And we've done that also in Asia, especially in Korea, South Korea, and in China. And just a couple of examples here. We've been doing that with the USDA ARS, which is our sister organization in US, but also in Agropolis in France, Heulich Institute in Germany, and Wageningen University in the Netherlands. This is just to illustrate this program. As I said before, we are basically in US and Canada and in Europe. Our coordination is in France, in Montpellier. But we also have another mechanism for cooperation overseas, what we call the joint calls. Basically, Embrapa negotiates with our potential partners in specific topics for our research teams to elaborate on single projects and submit to a joint committee between Embrapa and its partner organization. We've done that, and we've been doing that with BBSRC from UK, Agropolis from France, INTA in Argentina. And the idea is the sharing of data, sharing of people, biological material, and so on. And
sorry, Alexander, this is your 2-minute warning.
Okay, I'm just finishing. So we share funds. The funds are for local use. We don't transfer funds. And this is just a figure to show you. Basically, there is this exchange between the partner institution and Embrapa in terms of scientists, data, biological material, students, and so on. And we don't share financial resources. We don't transfer financial resources. Of course, if the partner institution wants to transfer money to Embrapa, it's welcome, but it's not the basis of this collaboration. This is just an example of what we've done with BBSRC, and we think it's a very interesting model for cooperation. So bringing together research centers, other universities in Brazil and research centers, and also bodies from UK. Another one is the visiting scientists programs. So we are able to place our researchers overseas for up to 1 year in world-class research institutions such as universities, research centers. just to give you one idea where we've been placing our researchers to foster cooperation with our partners. And the last one is the co-funded projects that we take advantage of our funding agencies in Brazil and overseas. It's we can do that through public and also private bodies. Open innovation is a big business for Embrapa nowadays, and we've been collaborating with private companies as well. So that was the last slide, and that was just to scratch the surface of this possibility of cooperation with Embrapa in terms of you know, a number of topics, including plant phenotyping. Okay, thank you very much for your time.
Thank you very much, Alexandre. And I want to invite to all participants to use the messenger system in the Cuba app to get in contact and ask questions and continue the discussions, in addition, of course, to the chat. Thank you again, Alexandre. Okay. Our next talk is from our colleague, Juan Miguel González-Aranda, who is the Chief Technological Officer for LifeWatch. Inmaculada already told you about 4 of the projects, the pilot projects, that are part of EU-LAC ResInfra, and LifeWatch is one of these pilot projects, and Juan is going to be telling us a bit more about it. Then, Juanmi, if you would like to share your screen.
Hello, good morning, good evening, bon dia, buenos días. So I want to share, but you need to allow me, please. Now, now I guess that I can. Yes, can you see my screen?
Almost there. Yes, yes, we can.
Okay. Let's put. Okay, now full screen. So thank you very much for this again magnificent opportunity, based on the fact that the cooperation between America, Latin America and Caribbean and the European Union is essential, and in particular on biodiversity and ecosystem research. Why? I am really happy to have checked that previous presentations, the high quality of the presentation of my colleagues from Brazil previous related to biodiversity, how one of them is related to genetics, how it is connected to biodiversity, and the last one with agriculture, agroecology, and sustainable development, with the Green Deal. Well, I will go in detail. I like to do connections so that we are going to show what is now the situation of our LifeWatch pilot case study. So, LifeWatch ERIC in a nutshell. LifeWatch ERIC, maybe you know, is the science European distributed infrastructure focused on how to measure the impact of global climate change issues on Earth, biodiversity and ecosystem research, as previously Immaculada said. The expected impact is to be a structuring tool for the European research area, also supporting policy decision-making, addressing society challenges which demand scientific knowledge in a global climate change context, including citizen science activities. Wow, how to put so many contents in only one paragraph, on one sentence. What does it mean? That we are trying to provide the state of art of ICT, big cloud data, artificial intelligence, deep learning, edge and HPC computing, high-performance computing, blockchain, in order to support communities, not only researchers, but also decision makers and citizen scientists. Well, from the point of view of the global biodiversity informatics outlook, defined for Donald Hoover et al. In 2012, they identify 20 components for biodiversity informatics. There are 4 layers: culture, data, evidence, and understanding. We are on the top from the point of view that we need to understand what is going on, based on scientific evidences. That is like what ERIC about. It's about multi-case species modeling, trends and predictions, modeling biological systems, visualization, dissemination, etc. And to that purpose, we have released, let's say, we are developing, co-developing in an iterative, incremental way 2 ICT, 2 software tools. One of them is the so-called Tesseract, that is the new generation of virtual research environments, and LifeBlock, that is a typical blockchain technology based on hyperledger permissioned system, but with one small innovation. The small innovation, let's say, is the tokenization of ecosystem services, how to put in value the preservation of biodiversity and ecosystem services in societal challenges or critical essential points, topics such as agroecology. So, to that purpose, the European Union, among the member states, had defined what you know, what is the ERIC, the European Research Infrastructure Consortium, based on the S3 roadmap. A LifeWatch ERIC is structured by a triangle composed of 3 common facilities based on the Netherlands, Italy, Spain, jointly with other observers and candidate countries. And we are not alone. We have a pan-European perspective, international perspective, connected, tightly connected with AGV, the Global Biodiversity Information Facility, Copernicus, Research Data Alliance. Wow. And I can be all day mentioning a lot of friends and colleagues working with us. So that you know better than me that there are ongoing from the past, currently, and there will be for sure in the future a lot of collaboration with Latin America and Caribbean. Here you can see only some names, because you know better than me that the 86% of meso and macro biodiversity is there. It's here, let's say, from the overall Latin America, Caribbean area. And we have made a very exhaustive study of what is going on. And as a consequence, we are key partners in ResInfra project. We are colleagues from jointly with Claudia and with Inma and so many friends there. From the point of view that after some field visits and some high-level political meetings, we have identified some key core groups in order to pave the way to that collaboration. Due to the importance of biodiversity and ecosystems in economic growth and equity in Latin America and the Caribbean. That is clear. I will not go through the technical analysis defining what is our pilot about, but what I can say is how we can support you as a research infrastructure, we as a distributed infrastructure to you from the strategic, tactical, and operational point of view. How to achieve these objectives? First, strategically, we need to put together the strategies of IPBES in relation to ecosystem services in accordance to the Sustainable Development Goals and the European Union Green Deal and Euro Biodiversity 2030 objectives. We need in some way to find a way in a tacit knowledge procedure to find the correspondence, the twin experience on that, based on that new socioeconomics model. This is high level politically oriented. It's based on the creation of high level.
Tenemos que crear, como ha dicho Inmaculada, grupos de trabajo de alto nivel que integren a investigadores, a legisladores y a la sociedad. Desde el punto de vista táctico, no reinventemos la rueda. Ya hay mucha colaboración. Nosotros ya hemos identificado estas colaboraciones y utilizamos herramientas de computación avanzada para identificar redes. Y aquí tenemos algunos ejemplos. La parte inferior derecha ven un nodo muy sofisticado. Estamos hablando de, bueno, nuestro sistema de blockchain que nos ha permitido identificar la colaboración.
In the blockchain, we can measure the intensity of cooperation among them.
Medir la intensidad de la colaboración entre las distintas partes. Ya sabemos dónde están, nos hemos mapeado. GeoBond es el grupo sobre observaciones terrestres. Nosotros estamos trabajando en el modelado, tenemos que ser capaces de trabajar conjuntamente de forma eficaz. Como ya les he dicho, nosotros hemos creado el comité EU-LAC, creemos que no hay que reventar la rueda y creemos que es necesario aportar herramientas de colaboración abiertas. Y aquí, en esta diapositiva, ven la intensidad del trabajo ya realizado y el nivel de implicación de los socios de América Latina y el Caribe. Del lado europeo, la colaboración emana de Portugal, España, Italia. Y cuanto más verde, más intensa es la colaboración de los socios. Estas son buenas noticias y pese al confinamiento y pese a la situación de la COVID-19, lo cierto es que seguimos vivos, seguimos colaborando y estamos ansiosos de seguir colaborando. Tenemos que consolidar nuestra...
Anyway, I would like to contact the previous speakers with Chile, with Colombia through the GBF International, mainly with Costa Rica, where we are almost to establish finally. We have established in a virtual way, but not finally in a physical place the Central American Caribbean LifeWatch Eric Green Hub, mostly focused on agroecology issues with the collaboration with the Inter-American Institute for Cooperation of Agriculture, with Cuba, not yet the start, but initiated, with Honduras, not yet the start, but in contact with the America's Flyway Initiative, Mexico, in a very nice, the so-called Eco-BioCal initiative, jointly getting together Argentina, Dominican Republic, Ecuador, Guatemala, Nicaragua on what is vivienda sustentable, sustainable living and ecosystems. Paraguay, we are in ongoing conversations in relation to Alianza del Pastizal. Peru, in relation to freshwater and passing ocean socioeconomic impacts. And Uruguay, last but not least, we are going to establish the twin in Uruguay, similar to Costa Rica, to be the co-located main office in a particular focus on innovation, on bioeconomy, blue economy, more related to oceans and marine ecosystems. This is our work plan. I used to say that many times we are used to create...
Sorry, this is your 2-minute warning.
Yes, I was finished, really, almost. So, thank you, Cassie. Well, these are the work plan. This is the timetable. You know that the Gantt and all these tools are used creative for not accomplishing. Well, we are with a little bit delay, we recognize, but we are accomplishing almost to the fitting to the different objectives. And that's all. And I would like to thank you, all the community, and please leave the spirit high. Mucho ánimo. We just almost there. We will meet together very soon, face to face. As apes, mammals we need to meet together very, very, very soon. You know, Claudia, very well, that I'm not optimistic, I'm enthusiastic. There is a small difference. And thank you very much. And we remain at your disposal. Thank you so much. Muchas gracias. Muito obrigado.
Thank you very much, Juan, and it's very optimistic as always. Thank you very much. And last but definitely not least, our last example will be brought by Chris Wood from the National Laboratory for Advanced Microscopy in the Universidad Nacional Autónoma de Mexico. And this is a great pleasure, as we have been collaborating with Global-BioImaging and other of the research infrastructures and the group from Chris Woods, and I'm really interested to hear what he has to tell us about their infrastructure. If you want to share your screens, Chris?
Yeah, I'm going to go ahead now. So I think you can see the presentation now?
Yes, we can.
Okay, it's just... there we go. Okay, thank you very much for the introduction. It's a real pleasure and honor to be able to present the bioimaging perspective from Latin America. I have quite a few slides, so I'm gonna crack through. In a historical context, the region has a long history of cooperation of academic societies that promote microscopy through all these different countries. Currently members of the organization of the Inter-American Committee of Societies for Electron Microscopy. Generally and historically, there has been more of a focus on electron microscopy in the region. And that's because electron microscopes have always been large, expensive, complex, and technically demanding. But really now advanced light microscopes are now in the same category. And such equipment require research instrument infrastructure to best support it. And so I'm really coming from a light microscopy perspective. That's where I'm gonna come from today. I think we all understand why research infrastructure. Why do we need it? Really, it's to make the best use of the greater capital investment and dedicated support infrastructure to have highly specialized human resources involved and to just open up access to these highly important facilities for all for driving research outcomes. So bioimaging research infrastructures, they're wonderful platforms. They're particularly in bioimaging, they're extremely multidisciplinary, integrating a lot of different disciplines into the experimental studies. And so, as we all know, research infrastructures are are great for building multidisciplinary collaborations, democratizing access to the technologies, to spark creative interactions and promote quality assurance, and to train current and future generations of scientists at the highest levels. And all this is really about people. It's really about a people interaction dynamic. This is about bringing people together and getting creative and allowing people to interact and spark new ideas and innovations. So research infrastructure is expensive and requires long-term support, and the degree of which we have existing research infrastructure in bioimaging is patchy across the region. So Argentina has a centralized and organized government support for microscopy since 2008. And also in Brazil, Mexico, Uruguay, and Chile, there have been specific funding schemes to support research infrastructure. But generally across the region, the support can be sporadic. It's not really regionally coordinated at the minute, and there are differing degrees of success in implementation. So can we look to where research infrastructure has a longer history of multinational support and integration? We certainly can, and that's what we've been doing. So Claudia mentioned RI-VIS, which has a much longer history of integrating these activities and supporting research infrastructure. This is a slide they passed to me. So RI-VIS is a distributed research infrastructure with nodes across Europe. They have 16 countries involved. They have over 100 sites actually incorporated within the RI-VIS umbrella, which is organized around 25, particularly around 25 nodes, which are centers of excellence, which have a particular status within the RI-VIS organization. Due to the great success, they're involved in a lot of different activities, too many to detail here, but due to the great success that they've experienced, Global-BioImaging was funded kind of as a spin-off from RI-VIS. It was a Horizon 2020 funded project, and initially it had a very good global reach around different regions around the world. Argentina was one of the first sort of founder members of the Global-BioImaging project, one of the early adopters of the initiative, and that's expanded and grown over the years, and currently there are a number of has grown to include pretty much all the Americas and other regions around the globe as well, pretty much covering all of the continents. So what are the activities that they're involved in? What is their contribution to the community? It's very much again based around human resource. It's based around and developing common structures and common standards for bioimaging around the world. So it's involved in staff exchange, job shadowing programs, working groups, thematic workshops, exchange of experiences, lots of organization of interchange of ideas, and developing, as I said, international recommendations for things like metadata, data exchange in the bioimaging community. And that community really is the key. So that contact, that communication side, the exchange of experience meetings, the last physical meeting was held in Singapore, have been key to developing contacts between different areas around the globe in the bioimaging community. Okay, now from a Mexican perspective, well, there's a lot of data here. I'm not going to go into it, but I represent the National Laboratory for Advanced Microscopy in Mexico. And since 2013, it's been expanding. We now have actually 3 sites across the country. That doesn't mean that we cover everywhere. There are a lot of other imaging centers, open access imaging centers, which are the ones that I know about are covered on this map. And we probably need to do much more work locally to integrate these resources into a community. But as the director of the National Lab, I've been since 2018 representing the Mexican bioimaging community in both Global-BioImaging and Bioimaging North America, which was an organization that grew out of a meeting organized at Geneva Farm in 2018. We're participating in the training, different working groups, and involved in interchange and networking with homologs across the planet. And these strategic alliances have been reinforced through the signing of memorandums of understanding to actually formalize the relationship. This has been quite an important element. So our take-home message from that participation is how extremely positive it's been. The notion that the issues that we're facing are really nearly universal, although their scale and priority can vary. Signposts and pathways towards solutions we can encounter those, and then we can adapt them to local conditions. We can strengthen the confidence of local bioimaging communities, overcoming energy barriers, And we can also contribute to the wider sector development and obtain agency and diversify initiatives. So the positive experience of the national microscopy in Mexico and its interaction with the global community helped catalyze a bilateral initiative between Mexico and Uruguay, which was it's not a science funding initiative, it's a foreign ministry initiative, very much based around social development, based around the Mexico-Uruguay Cooperation Fund. So this was together with two researchers in Uruguay, Andrés Camaid and Leonel Malacrida, between the Institute Pasteur and the Universidad de la República. And the title was a cooperative proposal to develop advanced imaging, optical imaging capacity with the aim of establishing a Latin American light microscopy network. There was very, very little science in this application. It was all about capacity building. It was all about using research infrastructures, building them and using them as platforms for social development and international cooperation. And I think they're ideally, that's one of the things I want to stress. I think research infrastructures are absolutely ideally positioned to achieve those goals. So what did we achieve? Well, there was an awful lot of meetings involved, but the pandemic has put that on hold. So we've been involved in more virtual events recently, but we did have some interchange visits, consultancy, reports written for establishing a national microscopy laboratory in Uruguay that was actually founded in 2020. And we are still have on hold a final event, which is an international meeting around the theme of promotion of integration of these resources and services in Latin America. And one of the things I already stressed, I wish to stress again, excellent research infrastructures and bioimaging are excellent vehicles for bilateral and regional development. They promote cooperation, educational, social, and economic development. So we've had events in schools promoting microscopy, not just at the research level, but also at social development level. And also a driver for regional integration. And that's something we've very much strongly learned in lessons from the European Union. So that has led to the founding of Latin America Bioimaging, which is just beginning its activities this year. So we had a first event, which was an introductory symposium in April 2021 with 130 international attendees. And the mission is aligned with many of the goals of the other bioimaging initiatives around the world: promote bioimaging to academic and political communities, develop and support capacity building activities in the region, build databases of bioimaging researchers and microscopy centers, and promote community integration and create a bridge to international partner organizations. And we have found partners identified in the following countries. And this brings us full circle. So now Latin America Bioimaging is an ideal platform to consolidate the interaction with the international global communities. That's where these initiatives have come sort of further and further from the ground up and are now starting to be coordinated across the region. So what are the conclusions from these experiences? Well, bioimaging research infrastructures are emerging and strengthening across Latin America. The collaboration between the European Union and Latin America is growing under the umbrella of Global Bioimaging Network, with the close involvement of EuroBioimaging, of course. And formalization of the collaboration is being achieved by an international memorandum of understanding. It's actually been a really important instrument. What are the perspectives and challenges? Well, one of the things we need to do is to bring this convergence between bottom-up efforts that start within the community, and to promote the kind of the top-down efforts that are also required, and to bring sort of strengthen and widen the base, distributed access to resources. How can we sort of use these infrastructures as a leveling up perspective across different regions which have disparate pre-existing investments in science infrastructure? Obviously, all the human resource side, training for research infrastructure management, managing research infrastructure is very different to sort of the traditional academic training that most academic scientists have undergone. So we need to improve that. We need to have mobility schemes to improve access to the resources and to ensure that we aren't unnecessarily duplicating resources in different areas when improved mobility would solve that problem. And we need to think about maybe generating nodes for high technology and training. Some of these technologies are so expensive, it would be a folly to establish many of them around the continent. From the top down, where we need to get the value of research infrastructures recognized as platforms of social development and international cooperation at the political level, we need to increase the multilateral coordination to maximize resource funding efficiency. We need to update funding and evaluation structures to better support research infrastructures and the human resources and their particular needs. They really don't fit necessarily into the sort of traditional funding structures that have been built up over decades. And we need to recognize
just a warning, time running out.
Yep, no problem. I'm very, very near the end. And their human resources. And we need to sort of better integrate different research infrastructure to avoid zero-sum conflicts, because we're in a very usually quite resource-starved region. So we need to integrate these research infrastructures to prevent basically conflicts occurring between over a scramble for limited resources. And we need to leverage those synergies. So in conclusion, we need to really set up this virtual circle. That's what we all want. So we need to start from strong bioimaging facilities, because strong bioimaging facilities will lead to strong national research infrastructures, and strong national research infrastructures will lead to strong multilateral integration. And for that, we need to get better integration between the communities. We need to get better coordination between the scientific, the government, and the inter and non-governmental organizations. This is critical. All of these, this will be a result when we manage to better achieve this central objective. Thank you very much for your attention. This is my email if you wish to get in touch. We have an email also for the Latin America Bioimaging Organization. Thanks to all of these groups who are coordinating these efforts, and other groups around the region. And if there are any questions, I've already been interacting in the forum. It's been great. Already posting messages there and interacting with the community here in this meeting, as well as in chat. So thanks very much.
Thank you very much, Chris. And I'm definitely going to start using the circle slide. It's very, very interesting and very useful. And it's also our experience of how things work. I want to thank all the talks again. They have been really interesting. And before I pass to Inma for final words to wrap up this very interesting first day, I want to remind you all that you can join us for a visit to the synchrotron, a virtual visit that will follow just in a few minutes when this session finishes, and to join us again tomorrow for our second day of the meeting. And as Chris said, to join the conversation in the messaging, what is being already very interesting.